Everyone. My name is Lian Pan. I'm a senior marketing manager at GeneScript. Originally, I invited Dr. Andy Liu, who is a senior scientist at our protein production department, to talk about the recombinant protein production in Baculovirus Insect cell expression system. However, Dr. Liu has a medical emergency, so therefore I invited his colleague, Keshav Vasasamvada, to give the presentation for him. To ensure the sound quality, all the attendees are muted. Please feel free to type in your questions in the question box in the lower right corner of your screen throughout the talk. Keshav will address your questions altogether at the end of the webinar. Without further ado, let's welcome Keshav to give his talk on the recombinant protein production in Baculovirus insect cell expression system. Welcome, Keshav.
Thank you, Leanne. Hello and welcome, everyone. Thank you for taking the time to attend this webinar today. As Leanne mentioned, I'll be the substitute presenter for Andy Liu today due to unfortunate circumstances. A quick background about me. I'm the marketing specialist for protein services at GeneScript, and prior to joining GeneScript, I worked for many years in the industry in a core facility doing molecular biology and protein biochemistry work, including expression and purification of a variety of disease targets to support the therapeutic areas on site. And so I have hands-on experience with the different expression systems, including insect, E. coli, mammalian, and yeast systems. And so in line with my experience, on behalf of Andy, I'll discuss certain aspects of recombinant protein production in a baculovirus system, insect expression system. During the webinar, I'll also highlight some of the services that will potentially match your needs and your downstream application. So with that, let's move on. So this is an overview of the presentation. First of all, we'll go through the available protein expression systems, and then we'll discuss about baculovirus insect cell system optimization, including how to manipulate the insect expression system, how you could improve protein production in this host, and also when to use the insect expression system. And then we'll take a look at some troubleshooting and case studies that our production team has developed. And towards the end, I'll introduce the relevant services at GeneScript. I hope this webinar will expand your knowledge on protein expression and purification techniques in the baculovirus insect expression system. So one of the most important considerations before embarking on a recombinant protein production campaign is the choice of expression host. Now, choosing the appropriate expression system, as you know, is very important to obtain the desired outcome. So let's first take a look at the most common recombinant expression hosts. Now, generally, recombinant proteins have been expressed in bacteria, insect, yeast, and mammalian systems. And now, given this kind of choice, how do you select which expression system to use? While there are various factors that go into the decision-making process, the nature of the target protein and its solubility, its functionality, the protein yield requirements, user experience, intended application, and the speed of generation are often some of the most important factors that govern that decision. And generally, E. coli system is the commonly used expression system because it's easy to operate, fast, and low cost to scale up. And with similar feature, protein expressed in yeast is also used. It has good post-translation modifications, and as higher eukaryotic systems, both the insect and mammalian cells have really good post-translation modifications, and they're more powerful systems for expressing soluble proteins and proteins in a bioactive form. And compared to the mammalian cell system, the insect expression system is less expensive and is easily scalable with virus infection. although its post-translation modifications are not exactly the same as that of the mammalian cells. And so this slide lists certain advantages of each of the major systems. And if you look further at some of the cons of each of the system, this slide summarizes some of the disadvantages. As prokaryotic expression system, E. coli has no eukaryotic post-translation modifications. So it's not suitable for difficult proteins such as large size proteins, and there is some incorrect folding also going on in E. coli systems. If you look at yeast, the post-translation modifications of the yeast system is, there are some issues with improper glycosylation and oftentimes excessive glycosylation. Both insect and mammalian systems have, pretty good post-translation modifications. They're a little bit more expensive than E. coli and yeast, and production time often takes relatively longer in insect and mammalian systems. If you look at insect cell expression, SF9, SF21 hi-fi cells are, popular hosts for expressing recombinant proteins. Now this slide lists the number of entries in the protein data bank by expression system as a percentage of the total number of chains within identifiable expression system. Now data here shows that E. coli is the top choice for protein expression in general, and insect is the top choice in eukaryotic expression system. Now this is due to the fact that insect system is relatively less expensive than the mammalian cell expression, and for difficult proteins it's more powerful than both E. coli and yeast. Now coming to the topic, let's take a look at aspects of Baculovirus insect cell system optimization to see how one can manipulate the insect expression system. And we'll review aspects of protein production and look at some case studies later on in the presentation. Now the most popular insect system is the Baculovirus insect expression system. So basically this uses baculovirus as a medium to infect the insect cells for recombinant protein expression. The system can be used to express genes from many different sources. It's been pretty good for genes from eukaryotes, including fungi and plants. And you could also express genes from bacteria and viruses very well in the insect system. And recombinant proteins have been produced both as fusion or non-fusion proteins at levels ranging between 0.1% all the way up to 50% of the total insect cell proteins. So about the baculoviruses, they belong to a diverse group of large double-stranded DNA viruses that infect a variety of insect cells. And the baculovirus, the principle is that the baculovirus genome is replicated and transcribed in the nucleus of the infected host cells. And the most extensively studied baculovirus strain is the ACNPV strain, the Autographa californica nuclear polyhedrosis viruses, which replicates in the nucleus of over 30 different insect cell lines, with the most common ones being SF9, SF21, and Teeny cells. Okay, so to give a quick outline, you would have a donor plasmid with the gene of interest within the donor plasmid that you could use for baculovirus construction and amplification. And then the insect cell is infected by baculovirus for protein expression. So it takes about 12 to 15 days, for constructing the plasmid and then infecting the cells and then amplifying it to produce baculovirus and then harvesting it and then using it for insect cell expression. It takes about another 2 to 4 days for infection. Now back-to-back baculovirus construction. Back-to-back is a very powerful method for baculovirus construction. It is an abbreviation from bacterium to baculovirus. That's what back-to-back is. Now, this system facilitates the rapid generation of recombinant baculoviruses. The steps involved include cloning the gene of interest into a recombinant plasmid vector, and depending upon the vector selected, your gene expression is controlled by either the polyhedron or the P10 promoter. Usually in this schematic you see a polyhedron promoter. The expression cassette itself is flanked by the left and right arms of the T7 transposon, which is useful for integration. And the donor vector is transformed to a special competent DH10 back E. coli cells, which contain the helper plasmid that contains the baculovirus genome. Now this transformation facilitates the transposition of the donor DNA along with the insert into the BACMED genome. And when this happens, homologous recombination takes place within the cell, and the recombinant BACMED DNA is produced. Now this recombinant BACMED DNA is extracted and transfected to adherent SF9 cells after about 3 to 5 days, and the P1 baculovirus is produced right here, as you can see in the schematic. And then you could use blue-white colony screening to pick up the recombinant BACMED, and further PCR for identifying the right clone, as you can see in this gel image. Now in this step, 3 kinds of materials may be stocked as the backup, including the recombinant E. coli strain. You could save it more than a year at -80 degrees. You could store the recombinant BACMED DNA usually at 4 degrees for up to 2 weeks, and then you can certainly store the P1 baculovirus for many months at 4 degrees. Now further baculovirus amplification can produce from P1 can produce the P2 and the P3 virus. As you can see, subsequent round of amplification, you increase the amount of virus, and generally the viral titer increases from 10 to the 6 to 10 to the 8 PFU, platforming units per ml after 2 generations. And the infected SF9 cells and the supernatant, they can both be used to evaluate for your target protein expression. And the right, if you see in this image, we see a comparison between the control on the left-hand side panel. You see there is no target expression on the right-hand side. You can see the expression of recombinant protein is pretty obvious. And the best part is that every generation of baculovirus could be amplified easily for scale-up production, and this is a key advantage of the system. So finally, when you have enough baculovirus, you can infect the insect cells with it for protein expression, and small volume culture can be produced in different sizes of flasks. Typically we can do anywhere between 40 ml and 10 liter in flask. And for large volumes, we can conduct recombinant expression using wave bags, anywhere between 10 to 100 liters using wave bags. After about 2 to 4 days of culture, cell pellet and supernatant are collected for protein purification. And some of the parameters here, insect cells are maintained at 27 degrees, and the cell viability should be more than 95%. Usually the infected cell density should not be more than 2 million. And another advantage over the mammalian cell expression is insect cells do not need carbon dioxide, and we need oxygen and pH. Ideal pH is between 6.1 and 6.4 for growth. Now, having seen that, let's take a look at how we can improve protein production in an insect expression system. First of all, codon optimization is very meaningful way, very useful method to improve protein expression and also decrease the cost of final cell culture. And the concept is pretty simple here. Since not all codons are used equally within a host, the major codons are those that occur in highly expressed proteins, and the rare codons are in genes expressed at lower levels. And when the codon usage of your target protein differs a lot from the average codon usage of the host organism, this could result in decreased expression. So when, excuse me, when you have your genes codon optimized at GenScript, we use our patented optimum gene software algorithm. So what it does is produces over 10,000 virtual candidate sequences by analyzing your sequence, and then a complex sorting matrix selects the best sequence for each specific customized request. And then our algorithm optimizes a variety of parameters that are critical to transcription, translation, and protein folding, and eliminates any codon bias. And I can tell you we have optimized thousands of sequences in all major expression systems using our codon optimization platform, and we found that the expression level of target proteins can be significantly improved by the use of this technology. So you begin with codon optimization, clone the gene in the donor plasmid, construction amplification, prepare baculovirus, infect your SF9 or SF21 cells or TNE cells, and then harvest and test for recombinant expression. So that's your workflow. So how do I choose the appropriate donor plasmid? So there are different plasmids for different purposes. So we've listed a few on the slide in this table. For the PFAS back, the first one, the first row here, there's no extra residue and could be easily edited for optional tag fusion. For the PFAS back HD, it has the his tag at the a 6X his tag at the N-terminal region, and it could be cleaved by using the TEV protease. The PFAST-back jewel, it has two multiple cloning sites within one vector, so essentially you could use it to express two different genes. You could co-express it. So one is controlled by the polyhedron promoter, and the other is controlled by the P10 promoter. So if you have a multimer, you could use this to co-express and co-purify. Sometimes the PDEST20 vector can also be used because it has a cleavable N-terminal GST tag. So these are some of the vectors that you could use. You're looking at the expression strategy. So when you're using intracellular expression in addition to the his tag and GST tag, you can add a few other kinds of tags at the N-terminal and or C-terminal gene of interest. So you could try his GST FC flag. Typically a TEV region is added at the N-terminus, so you could use it to cleave off the tags and separate it from your recombinant protein. When you're looking at secretory expression, you could try a variety of signal peptides at the N-terminus. So you could try GP67, the HPM, GP64. And as you probably know, GP67 is the most commonly used signal peptide, and I listed the sequence here. If you want to express a complex with multi-subunits, generally we can use the PFAST-back dual vector to clone two genes into one vector. So both the genes can be expressed using just one vector simultaneously. Or you could also try one gene per baculovirus, and you could add multiple baculovirus to one flask, for expressing a complex, multiple proteins in a complex. And so this can also be a efficient way to express and purify proteins that are part of a larger complex. Excuse me. So optimization for expression. So what expression conditions can we optimize? So depending upon your application and gene of interest, you may use any insect cell line, including the SF9, SF21, Hi-Fi, or mimic SF9 for expression. Cells can be grown in either adherent or suspension culture in the culture vessel of choice. And this table here lists some of the 4 major cell lines. The doubling time for each cell line, whether virus amplification is needed or not, and the protein expression, whether it can be used or not. So if you're expressing a secreted protein, we have found that you can improve expression by using the Hi-Fi cells. And the optimal MOI, multiplicity of infection, will vary between cell lines and the relative infection kinetics of the virus isolate or clone used. We recommend that dose response be established for each virus, medium, reactor, and cell line employed to determine the optimal infection parameters to use for protein expression. Generally, as a starting point, you could infect cells using an MOI between 1 and 5. Plus fresh virus should be key for protein production. Generally, if it's older than 3 weeks, we recommend within 3 weeks is good. Beyond that, you could see a depletion in expression. So a tip for this is you can passage virus once for fresh application from P3 or even from P1 with more virus seed and more generation. As I said, we recommend performing a time course to determine the expression kinetics for recombinant expression. As many proteins can be degraded by cellular proteases that are released in cell culture. And maximum expression of secreted proteins is generally observed between 30 and 72 hours. And for non-secreted proteins, it's between 48 and 96 hours post-infection. And you could also supplement the culture post-infection with 0.1 to 0.5% FPS or BSA to protect the recombinant protein from proteolysis. Although we generally culture cells in serum-free conditions. At last, cell viability is important for good expression of your protein of interest. You should update the cells with fresh seeds at least every 4 to 6 months. Excuse me. The third point is, what can we do with this powerful expression system? So let's take a look at some of the published case studies and some of our own in-house case studies. Now, this here is some really great work expressing recombinant proteins using the insect expression system. Now, on the left-hand side here on the top, what you see is the influenza virus polymerase, which has 300 kilodalton subunits. The 3 subunit genes were randomly cloned in one vector and co-expressed in Hi-Fi cells for intracellular expression. The polymerase was recombinantly expressed in insect cells as a self-cleaving polyprotein. At the N-terminus, it encodes the tobacco H virus, TEV protease, that cleaves the C-terminal to the amino acid right here, ENLYFQ, and then releases the N-terminally his-tagged PA, PB1, and the C-terminally strep-tagged PB2, and the cyan fluorescent protein, CFP, which you see right here. And this is for the monitoring of expression. So you can see on this gel images, the protein is wonderfully expressed. On the right-hand side, in a separate study, you see the expression of the Huntington protein. It's a very large 350 kilodalton protein that was successfully expressed and purified in SF9 cells for intracellular expression. And because there was a chaperone protein in mammalian cells for Huntington protein, an additional step was needed for further purification. Actually, this was tried both in insect cells and mammalian cells, and the outcome in insect cells is much better. As you can see on the left-hand side, on the right-hand side, you can see the mammalian cell expression. There is some co-expressing chaperone proteins, which resulted in not successful purification. Additional steps had to be carried out. And going further, so here what you see is the case study of the T cell receptor. It has two subunits, the alpha chain and the beta chain. They were expressed in HiFi cells for secretory expression. Excuse me, the glycosylated site was matured well here. And this on the right hand side is a case in-house protein. We call it XX to retain the confidentiality of our client's project. So this is a 100 kilodalton protein. It was very difficult to express using other expression systems. So it expressed really well in our hands using HiFi cells. You can see very robust expression on this gel images, and we had a very happy customer who gave us a very positive testimonial. The protein generated by GenScript is 3 times more active than that produced by the academic, so the academic collaborator. So please pass my thanks on to your team. So a very happy customer, that used the protein in a functional assay downstream. So these are a few examples. You can also produce kinases, cytokines, membrane proteins very successfully using the insect expression system. So now let's take a look at the services that GenScript provides that can match your needs. So if you take a look at this table here, so we have a variety of packages. We have a small scale expression evaluation optimization package called the Potential. So here we can try one subcloning strategy and do transfection. And the deliverables include an expression evaluation report, and it's a very competitive package that starts at $400. We have customized insect expression packages that start from gene synthesis, subcloning, wild generation or amplification. If you're interested in such a service, you can inquire a quote for this. It's charged based on the services that you request. The quote is free of cost, by the way. We have guaranteed insect expression packages. We try different subcloning strategies. We can deliver 1 milligram of protein between 75 and 85% purity. It's a very competitive price. It's $3,950. And this is a fail-safe guaranteed package, because if we do not deliver the protein as promised, then there is no charge to the customer. So this is a really great package. We have the frag power guaranteed package for expression of fabs. We co-express the light and heavy chains of the fab region, and this is about $3,399. We deliver 3 milligrams of fab and greater than 90% purity. This is another example of a great guaranteed package. And we have different packages that can produce the baculovirus stocks for you. We try one subcloning strategy. We prepare the baculovirus, excuse me, and do the expression evaluation. And over here, we list the prices for each of the packages. It's about $1,900, $2,250, and $2,700 for the baculovirus at different PFU per ml, different titers. So, excuse me. Now, looking at GenScript's experience, the left side chart shows you the number of proteins that we expressed in bacterial, yeast, insect, and mammalian cells, and the right side pie shows the percentages of different types of proteins that we successfully produced with GenScript. So you can see we produced a variety of proteins very successfully, including kinases, cytokines, antibodies, antibacterial peptides, and so on. And so far, GenScript has delivered over 5,000 proteins in 4 different expression systems, and we have a pretty good success rate. It's about 95% for all protein projects. So no matter what kind of protein production projects you have, you can find a solution here at GenScript. We offer a variety of protein services covering almost every aspect of recombinant protein expression and purification, from transient expression to stable cell lines, from protein expression evaluation and optimization to large-scale protein production, both upstream and downstream process development, and GLP-compliant bioprocess services. We have very rich experiences on recombinant antibody production services, so we can successfully deliver between milligram to gram level recombinant antibodies. Excuse me. We also have a very large capacity for high throughput production for both recombinant antibodies and protein variants. To give an example, for protein variants, we can produce over 1,000 protein variants in just 30 days using our high throughput protein variant service. And for some really difficult protein targets, we can produce them using our advanced chemical protein synthesis services right here, it's listed. And we also have extensive technical resources and bioinformatics tools listed on our website. So I recommend that you check out our website, genscript.com, and go to the protein services section. You can find a variety of tools that can help you accelerate your research. And so in wrapping up, this is a slide that lists some of our other protein services besides protein services. Our other services besides protein services, we list gene, peptide, antibody, cell line, and discovery biology services. All this with the goal to make your research easy. And we also have more than 13,000 peer-reviewed journal articles that have cited our services and products, making us the most frequently cited biology CRO in the world. And this is a slide that lists our production team scientist, Andy Liu's email information. So if you have any questions that are not addressed at the end of the session, you can certainly reach out to Andy, shoot him an email, and he'll get back to you. And with that, we reach the conclusion of this presentation. Before we go to the Q&A, I want to restate that this webinar, along with all our other earlier webinars, will be archived on our website that is shown here, right here. So please visit our webinar page and take advantage of these resources. So with that, I guess we can move on to the Q&A. If you could just give me a few minutes, I'll just scroll and see if we have any questions that came in. So, excuse me. So we have one question, SF9 versus SF21 versus Hi-Fi, which cells are better? So that's a good question. SF9 cells are known to be a subclone of the SF21 cells. They were chosen because of their faster growth rate and relatively higher cell density than SF21 cells. Now, depending upon the protein that's expressed, all three cell types can compare favorably in terms of heterologous expression. So essentially, it's all protein dependent. In some cases, for example, in case of secreted proteins, sometimes we find Hi-Fi cells perform better, and in case of intracellular proteins, SF9 usually perform better. But then again, it's all protein dependent at the end. What's the largest protein that can be comfortably expressed using the baculovirus system? So that's a good question. We have seen in our case studies proteins as large as, for example, in the case of Huntington protein, it's a protein that's 350 kilodaltons. it has been successfully expressed. Now, I do not know what the upper cutoff limit is, but you can express really large proteins. I can check with the production team and we can get back to you on that. So using insect expression, what kind of applications can I use recombinant protein for? So you can use recombinant protein expressed in the insect system for a variety of applications. Typically, we see researchers doing subunit vaccine studies use this expression system a lot. You can produce kinases very successfully that you can use in kinase assays. You could use them for structural biology applications and other activity assays and drug development assays. So if you just give me a few more minutes, I can. So how do we store the baculovirus solution? Typically, it's stored at 4 degrees. I wouldn't recommend storing at -20 or -80. For a few months, say 3 to 6 months at 4 degrees, you expect good titer. You can still reconstitute and use for infection. If using a signal peptide to secrete protein, a signal peptide feed off upon secretion. Yes, upon secretion, you shouldn't. It's processed by the cellular machinery, so you wouldn't see the signal peptide in the secreted protein. Are there major changes in protein expression if the virus is older than 2 months or kept in fridge? It varies case by case. Now, I have seen, I have used virus very successfully for infection even after 6 months, and I have seen pretty good expression. In other cases, in about 2 or 3 months, I see there's a decline in expression. So in the end, it's case by case, but we would recommend best practice would be about 3 to 4 months at 4 degrees. 4 degrees, I wouldn't go longer than that. Okay, so there are a few questions, one about glycan differences that I'm unable to answer. I'll funnel that to our production team. So I guess, yeah, with that, there are a few questions that I'll shoot ahead to the production team and make sure we get back to you. I guess with that, I guess it's time to wrap up our webinar today. Again, I sincerely do appreciate your participation and all your thoughtful questions. Thank you for all your time and kind attention. And on behalf of the entire team at GenScript, I wish you all success in your research. Thank you.
Thank you, Kishan, for the nice presentation. So for those questions we did not have time to address here, so we will follow it up by email. So you will get the answer with your questions. Thank you.